Hello everyone, I hope you are well. In this session, I'm going to talk about the cell membrane potential. Okay, the electrical difference or voltage difference between the two sides of the cell membrane, which is due to the difference in concentration of anions and cations on the two sides of the cell membrane, called membrane potential. The before session I said that we have a different concentration of anion and cation in the inside and the outside of the cells. Okay, I told you, for example, the sodium and the chloride concentration is higher than to the extracellular fluid, and we have a high concentration of the potassium in the inside of the cell membrane. So this difference in the concentration of anions and cations name membrane potential. We have three types of membrane potential in the cells. Include resting membrane potential, in brief RMP, and local or graded potential or electrotonic potential, and the action potential. The role of ions and important channels in membrane potential are sodium or potassium ions. We have a sodium-potassium ATPase pump in the cell membrane that is actively transport 3 sodium out and 2 potassium into the cell, maintaining the unequal distribution of these ions across the membrane. And the sodium channels allow rapid influx of sodium during the depolarization phase of the action potential, and the potassium channels allow potassium to flow out of the cell and contributing to the negative resting membrane potential, because the cation exceed or flow out in the cell, and we have a repolarization phase. The other channels that important channels in the membrane potential are potassium leak channels. These channels are about 100 times more permeable to potassium than sodium, allowing potassium to constantly leak out of the cell. This outward flow of potassium in the primary contributor to the negative resting membrane potential around minus 50 millivolts in the neuron in the cell in the central nervous system. And the chloride channel can also contribute the resting membrane potential dependent on the cell type. For example, in the CNS, the chloride has not effect in the or has not role in the resting membrane potential. And the calcium channel through voltage calcium gated calcium channel can trigger various cellular processes such as neurotransmitter release or muscle contraction. In briefly, the interplay between the selective permeability of the membrane to different ions, the activity of ion pumps and transporters and the opening or closing of specific ion channels are the key determinants of the resting membrane potential, and they generate the action potential in excitable cells. Okay, let me more explain about the resting membrane potential, or RMP. The resting membrane potential is the electrical potential difference between or across the cell membrane when the cell is at rest without any stimulation. Okay, this potential difference is generated and maintained by the selective permeability of the membrane to certain ions, particularly potassium and sodium, as well as the active transport of these ions by the sodium-potassium ATPase pump. You must know we can record the resting membrane potential in the all of the live cells. Okay, the resting membrane potential varies between the minus 100 to minus 10 millivolts in different cell type. For example, in the red blood cell, the resting membrane potential is minus 9 to 10 millivolts, and the epithelial cells is minus 18 millivolts, and the hippocampus cells in the CNS, for example, is minus 18 millivolts. Okay, as a whole, in your book, all of the neurons, the resting membrane potential is approximately minus 70 millivolts. And the key player involved in establishing the resting membrane potential are sodium, potassium, ATPase pump that maintains the unequal distribution of sodium and potassium across the membrane, and potassium leaky channels. So in the resting membrane potential, the potassium ions is very, very important, and the sodium ions have not role in the form a resting membrane potential, because the potassium ions are about 100 times more permeable to the than sodium, and allowing in the potassium leaky channels, allowing the potassium to flow out and contribute to the negative resting potential, because in this channel, in the resting membrane potential are open, so the potassium as a cation is flow as a cation flow out from inside to outside of the cells. So the this action can cause the negative in the inside compared to the outside of the cells. As a brief, for recording a resting membrane potential, we need a 2 electrode. A 1 electrode input to the cells and another electrode located in the outside of the cells in an extracellular fluid, and these electrodes connect to the voltmeter. In this voltmeter, in the resting membrane potential in the live cells, show the voltage difference between inside to the outside of the cells, and these voltage is a negative number. Okay, I told you we have, as you see in this table, show the resting membrane potential in different cell type. For example, in neurons, I told you this resting membrane potential is a minus 70 millivolts. For example, in the hair cell is a minus 40 millivolts. In the cardiac muscle cells is minus 90 millivolts, and the other type of the cells show a difference voltage between inside to the outside of the cells in a resting membrane potential. So, as a brief, the three factors contributing to the generation of the resting membrane potential, okay? The resting membrane potential was the voltage difference between the inside and outside of the cells in the resting situation. The one important factor that contributes to the generation of the resting membrane potential is unequal ion distribution inside and outside of the cells, and the existence of the leaky potassium channels always open, and the permeability of these channels to the potassium is 100 times more than to the sodium, and the inability of the most ions, for example the potassium ions, is trapped in the inside of the cells, and these ions cannot leave the cells. So these ions can cause the negativity of the inside of the cells compared to the outside of the cells. And another factor that's important to form of the resting membrane potential is sodium potassium pump. I said about this pump in the before session. Okay, we have three key terms related to membrane potential changes are depolarization. The depolarization refers to a decrease in the negative resting membrane potential, making it less negative, okay? This occurs when there is an influx of positive ions such as sodium into the cell, reducing the difference in electrical charge across the membrane. And depolarization is the first phase of the action potential. I will explain about the action potential in the other slide. The action potential is where the membrane potential rapidly becomes positive. The other terms is repolarization. The repolarization is the process of restoring the negative resting membrane potential after the depolarization phase of the action potential. This happens when the voltage-gated sodium channels close and voltage-gated potassium channels open, allowing the potassium to flow out of the cells. And the outward movement of the potassium ions restores the negative charge inside the cell compared to outside. And finally, the hyperpolarization refers to an increase in a negative resting membrane potential, making it more negative than the normal resting level. Okay, this can occur due to the activation of the potassium channel or chloride channels, which allow these ions to flow out of the cells, further increasing the negative charge inside. Hyperpolarization makes the cell less likely to generate an action potential, as the membrane potential is driven away from the threshold. As a summary, the depolarization is the decrease in the negative resting membrane potential. Repolarization is the restoration of the negative resting potential, and hyperpolarization is an increase in the negative resting membrane potential beyond the normal resting level. And the majority related to the slow kinetic of the potassium channel. So when the closing potassium channels is long, so we have a hyperpolarization. Remember that at rest, the inside of the cell has a negative charge, while the outside of the cell has a positive charge. I talk about the membrane potential. I talk the membrane potential in the cells include the resting membrane potential, local potential, and action potential. I talk about the resting membrane potential in a before slide. In this slide, I'm going to talk about the local potential. Another name of the local potential is graded potential, is generator potential, is electrotonic potential. These potentials are small, localized changes in the membrane potential that can cause depolarization or hyperpolarization. So you must know the resting membrane potential can be recorded in the cell at rest, but the local potential and the action potential, we need a stimulus for recording them. The local potentials can arise spontaneously due to intrinsic properties of cells, e.g. in pacemaker cells of the heart and some smooth muscle cells, and in response to a stimulus that alter the membrane properties, e.g. sensory, touch, or chemical stimuli. In this part, the local potential called the receptor potential. Okay, I will talk about the receptor potential in the nervous system physiology. The local or graded potentials features are the magnitude of the potential change depend on the strength of the stimulus. So the other name of the local potential is graded potential. So these potentials have graded response, and they do not follow the principle of all or none, and these potentials propagate over a short distance with a decremental conduction, and they exhibit an spatial and temporal summation. So in this picture show the EPSP and IPSP. EPSP is excitatory postsynaptic potential, and IPSP is inhibitory postsynaptic potential, are part of the local potentials that are generated in the postsynaptic membrane. Okay, EPSP it is generated when a presynaptic neurons transmit an excitatory stimulus to the postsynaptic neurons. It causes a temporary increase in a postsynaptic membrane potential and increases the likelihood of its excitation. But and the right graph show the IPSP. The IPSP, it is generate when a presynaptic neurons transmit an inhibitory stimulus to the postsynaptic neurons. These stimulus can cause the more negative charge in the postsynaptic neurons, and it causes a temporary decrease in the postsynaptic membrane potential and decrease the likelihood of its excitation. These local potential are generated in the postsynaptic membrane and remain localized without propagating as the action potentials along the membrane. In fact, APSP and IPSP play an important role in determining the probability of the action potential generation in the postsynaptic neuron. The another type of the membrane potential is action potential. Action potential is a rapid and transient change in the electrical potential across the cell membrane, which occurs in response to a stimulus. As you see, this action potential have some phases. The first phase named a fast depolarization phase, and then a repolarization phase that brings the potential back to the resting level, and a brief hyperpolarization phase. You must know the occurrence of an action potential requires the summation of a local or graded potentials to reach the threshold level, and the amplitude of the action potential is constant and does not depend on the strength of the stimulus, and the duration of the action potential varies depending on the cell type, typically ranging from the 1 to 3 milliseconds. In the previous slide, I mentioned the term of the threshold. You must know the threshold potential is the minimum membrane potential required to trigger an action potential in neurons. In this potential, suddenly the many number of the sodium voltage gated channels are open. I emphasize the action potential are initiated according to an all or none principle. Either the polarization does not reach threshold and triggers no action potential, or it reaches threshold and elicits an action potential that is always the same. In this graph, show the change of the potential or membrane potential related to the threshold stimuli. As you see, the subthreshold stimuli produce a sub-threshold potential or local potential, but the threshold stimulus produce action potential, and the supra-threshold stimulus cannot change of the amplitude of the action potential. And the I told you when the potential reach to the threshold elicit an action potential, and that is always the same, and don't change in the amplitude of the action potential according to the amount of the threshold stimulus. I told you two important voltage-gated channels have a role to produce action potential: sodium voltage-gated channel and the potassium voltage-gated channel. The sodium voltage-gated channels produce a raising phase of the action potential, and potassium voltage-gated channels produce a repolarization phase of the action potential. This picture shows the model of activation of these channels. As you see, the sodium channels in the above figure show the sodium channels. Okay, as you see, the sodium channels have two gates, activation gate and inactivation gate. As you see in the resting membrane potential, for example, in minus 90 millivolts, the activation gate are closed, but the inactivation gates are open. But any ions don't entrance to the cell from outside the cells to the inside of the cells. But when for example, sodium channels are activated by a depolarization of a membrane potential, when the membrane reach a certain threshold, for example, as you see, the minus for example, minus 50 millivolt, between the minus 90 millivolt to plus 35 millivolt in the depolarization situation, these channels open quickly. This function allows sodium ions to rush into the cell and leading to rapid depolarization, and which is critical for initiation of an action potential or the raising phase of the action potential. We have in these channels quickly inactivate, means the inactivation gate after a milliseconds, okay, very fast, the inactivation gates are closed, and so in this situation the activation gates are open but inactivation gates are closed. So in this situation the sodium again cannot entrance to the cells. As I told you, shortly after activation, the sodium channels undergo inactivation. This is a time-dependent process where a portion of the channel protein closes and preventing further sodium influx. And the inactivation is crucial for the propagation of the action potential, ensuring that the signals is unidirectional and allowing the cell to rest its membrane potential. As you see in the below figure, show the potassium channels. Potassium channels just have one gated. These gated are located in the inside surface of the cells that just have an activation gate. In the potassium channel active more slowly than sodium channel. They begin to open as the membrane depolarized, but reach full activation during the peak of the action potential. And the top of the action potential, the full activation of these channels allows the potassium ions to exceed the cells and contributing to repolarization phase of the action potential. I told you unlike sodium channels, potassium channels do not have a fast inactivation mechanism, and they remain open for a longer duration during the repolarization phase. And the delayed closure of potassium channels helps to stabilize the membrane potential and prevent excessive depolarization. And this slowly kinetic of the potassium channel can cause to produce after hyperpolarization. So as a summary, the sodium channels have two gates, activation gate and inactivation gate, and the kinetic of the sodium channels are rapidly activated and rapidly inactivated, and these kinetic allowing for the quick depolarization of the cells. But the potassium channels activated more slowly and just one gated, that's named the activation gate of the potassium channel, and can remain open longer and produced after hyperpolarization or hyperpolarization. And the activation of the potassium channel is reached to high in the top of the action potential curve, and this activation can cause a repolarization phase. The propagation of action potentials is a fundamental process in the functioning of neurons, and it is essential for the transmission of signals throughout the neuron system. As you see, the action potential propagates along the axon through a process called electrotonic conduction and the saltatory conduction. The continuous conduction in unmyelinate axon, the action potential propagates continuously along the membrane. Each segment of the axon undergoes depolarization and repolarization is sequence. This is relatively slow and requires more energy. But in the myelinate axon, the action potential jump from one node of Ranvier gaps in the myelin sheath to another. This name of the conduction is saltatory conduction. Myelin acts as an insulator, preventing ion flow across the membrane in the myelinate regions, which speeds up the conduction of the action potential significantly. This allows for rapid transmission of signal with less energy expenditure. The factors affecting action potential propagation are axon diameter, myelination, and the temperature. In an axon diameter, the large diameter axons have lower resistance and can conduct action potentials faster. And the myelination, I told you, the myelinated axon conduct action potential more quickly than unmyelinated ones. And temperature, the higher temperature can increase the speed of the action potential propagation. As a conclusion, the propagation of the action potential is crucial for communication within the nervous system. Understanding this process provides insight into how signals are transmitted in the body, influencing everything from muscle contraction to relaxes and sensory perception. The refractory periods are critical phases in the action potential cycle of neurons that ensure the unidirectional flow of electrical signals and limited the frequency of action potentials. They are divided into two main types: the absolute refractory period and the relative refractory period. The absolute refractory period is the time during which second action potential cannot be initiated regardless of the strength of the stimulus. This period occurs during the depolarization and early repolarization phases of the action potential, typically lasting about 1 to 2 milliseconds. The mechanism of the absolute refractory periods are during this phase, voltage-gated sodium channels are inactivated phase after they open during the depolarization phase, and the inactivation gates of these channels close, preventing any further influx of sodium ions, which is essential for the generation of another action potential. Because of this inactivation, no matter how strong a subsequent stimulus is, the neuron cannot fire again. The absolute refractory period ensures that action potential are discrete events and do not overlap, allowing for clear signal transmission. And it also sets a limit on the maximum frequency at which action potentials can be generated, which is crucial for the proper functioning of the nervous system. But the relative action potential, relative refractory period is the phase following the absolute refractory period, during which second action potential can be initiated, but only by a stronger than normal stimulus. This period lasts longer than the absolute refractory period, typically several milliseconds to a few hundred milliseconds, depending on the type of neuron and its conditions. During the relative period, some voltage-gated sodium channels have returned to a state where they can be activated, but many are still inactivated. And voltage-gated potassium channels remain open, allowing the potassium ions to continue existing in the cell, which makes the membrane potential more negative or hyperpolarization. And because the membrane is hyperpolarized, a stronger stimulus is required to reach the threshold for firing another action potential. The relative the relative refractory period allows for the possibility of a second action potential, but ensures that is less likely to occur unless there is a significant increase in stimulation. And this period contributes to the overall timing and frequency of action potentials, influencing how signals are processed and transmitted in the neural circuits. As a summary, the absolute refractory period, no action potential can occur, lasted about 1-2 milliseconds, and caused by inactivation of sodium channels. But the relative refractory period, a stronger than normal stimulus can trigger an action potential, lasted longer than the absolute refractory period, and caused by the reopening of the sodium channels and continued efflux of potassium ions. So refractory periods are essential for the proper functioning of the neurons, ensuring that action potentials are generated in a controlled manner. They play a crucial role in maintaining the integrity of signal transmission and preventing the possibility of backpropagation of action potentials, which could disrupt communication within the nervous system. Understanding this period is vital for grasping how neurons operate and communicate in both health and disease. You must know the shape of the action potential in the different cell of the body is different. Okay. For example, as you see, this picture shows the action potential shape in the cardiac muscle. In the cardiac muscle, the action potential have 4 steps. Okay, the 0 step, the rapid sodium voltage gated channels are open, and the top of this step, these channels close suddenly, and the transient outward potassium channels are opened. You see the voltage is decreased because the some potassium exit from the cell and flow out the cells. And after these type of the channels transiently closed, and we have a step 2. The step 2 is a plateau. Okay, in this step, the long-lasting calcium channels are open, and the potassium delayed rectifier potassium channel are open. And in this phase, the entry of the calcium ions balance the exit of potassium ions in terms of electrical charge, resulting in the membrane voltage remaining nearly constant. At the end of the phase 2 of the plateau, the long-lasting calcium channels closed, but the delayed potassium channels remain open until the end of the phase 3. After the each action potential, sodium-potassium ATPase pump becomes active and reestablishes ion gradients across the membrane. This requires the ATP breakdown, which produces heat in a neuron. As you see in this picture, the x-axis show the impulse per second, and the y-axis show the heat production. After the rest, you see the heat production is increasing. Thank you for your attention. Take care of yourself. I hope to see you in Iran as soon as possible.